Well, first I want to say thank you, Bacon, for the invitation, and it's a pleasure to be here and share my introductory fellowship work with everyone here. So, let me see. Okay, so we know that the sarcomere is the smallest functional unit in a striated muscle, and it's composed of thin and also thick filaments that all together they work to form this elegant and well-coordinated structure responsible for muscle contraction. Unfortunately, in some cases, alterations in the function of the sarcomere caused by, for example, variants in the sarcomeric proteins, they can lead to cardiomyopathies or disease of the heart, such as dilated cardiomyopathy and hypertrophic cardiomyopathy or AGCM. So in this schematic of the sarcomere, we can see that the majority of the sarcomeric proteins are associated with hypertrophic cardiomyopathy. And in special interest, I want you guys to take a look at this area that I highlighted in red, where you can see that all three troponin subunits, C, I, and troponin T, are linked to AGCM. And from now on, I will be focused on cardiac troponin T. So here is the cryo-EM structure of the native cardiac porcine thin filament. And we can see in blue the troponin T, which is the largest troponin subunit and is responsible for anchoring the troponin complex to the thin filament, among other roles. And the C-terminal domain is located in the core of the troponin complex. And the N-terminal domain, or TNT1, extends across the thin filament like a tail. So previously, our group published a meta-analysis study that concluded that TNT1 is a hotspot for pathogenic variants. And we show here the relative frequency of variants in troponin T mapped against amino acid sequence, location, and domain. And compared to other regions of troponin T, there is a high frequency of variants in troponin T N-terminal domain. And in general, the variants in cardiac troponin T carry a poor prognosis for the patient and are associated with high risk of sudden cardiac death. Now, being a little more clinical, back in 1995, a study was published where 112 patients from 11 families were screened for mutations in sarcomeric proteins. And 9 members of these families, they demonstrated a normal allele and a mutated allele with a TA transversion at residue 284. So there was a codon change from ATC to AAC, replacing then the isoleucine with an asparagine at position 79, causing the I79N variant. And sadly, when we look at the survival of these patients carrying the I79N variant along the years, compared to other variants in troponin T, we can see that they are expected to live around 35 years only, unfortunately. Now looking back to the structure of the cardiac thin filament, we don't see much detailed description of the N-terminal domain. We don't even see the location of the isoleucine 79 in this recent structure. Well, that's until recently, where Dr. Galkin and Dr. Christina Ricci, in collaboration with our group, they improved the structure of the native cardiac thin filament. Now we have a near atomic resolution cryo-EM structure of the loop region of TNT1. So we can see the location of 13 amino acids going from valine 75 until phenylalanine 87. And I would like to point out that with this improved structure, we observe the isoleucine 79 making hydrophobic interaction with methionine 355 present in actin backbone. So in addition to this I79 methionine 355 interaction, the loop region of TNT1 interacts with the actin backbone via ionic bonds, as we can see here in panel B, and also other hydrophobic interactions that we can see in panel C. And they stabilize the relaxed off state of cardiac thin filament. So we propose that the TNT1 I79 interaction with methionine 355 in actin stabilizes the cardiac tail domain and maintain the inhibitory property of the cardiac thin filament. So our hypothesis is that the presence of this I79 variant could then disrupt the interaction between TNT1 loop region and actin. And as a consequence, it will lead to the destabilization of the intrinsic inhibitory property of the cardiac thin filament. So for us to test our hypothesis, we use this transgenic animal model where we have a non-transgenic mouse as our control and a transgenic mouse carrying the human TNT I79. And the incorporation of the human mutated troponin T was 52%. And from these animals, I used the left ventricular papillar muscle to perform a series of experiments that I will show here the results now. So first, I permeabilized the cardiac muscle preparation because that would allow me to control the intracellular calcium levels. And then once I clip this tissue, I can put in a muscle rig apparatus and I can expose these samples to a range of calcium concentration going from low calcium to high calcium and measure a series of contractile parameters. So the first thing that we observed was that the I79N displays an increase in myofilament calcium sensitivity at both sarcomere lengths tested. I'm talking about 2.1 micrometer sarcomere length and also 2.3. And that's because we observed a leftward shift in the PCA force curve at 2.1 and 2.3. So now that we know that the cardiac thin filaments are not properly inhibited because of the mutation, and there is an increase in myofilament calcium sensitivity, we thought that more myosin binding sites would be available, which could favor cross-bridge formation and force production even at low calcium levels. So when we look at the force levels, we saw that the I7N myocardium displayed an increase in force generation even at low calcium levels of activation, and especially at the physiological range of calcium. And that's in both sarcomere length tested, as we can see in this highlighted area here. So next, we tested whether this variant is increasing active force even at below diastolic levels when the muscles will be fully relaxed. So for that, we performed KTR, which is kinetics of tension redevelopment at PCA 8. So during this experiment, what I did, I released the muscle 20% of its original length, and I quickly restretch the muscle 25% of the original length. That would break all the cross bridges that are then formed. And once I return this muscle to its original length, we can see the force starts to redevelop. So here you can see the force being redeveloping in every single sarcomere length tested, and you can see that the mutant the mutant myocardium is displaying an increase in active force at low calcium compared to the control at both sarcomere lengths. And we can also see that as we stretch the muscle, there is an increase in active force at low calcium levels within the genotypes. And that's truth for both our control and the mutant. So back in time, 23 years ago, Dr. Potter's group published this paper where they found that the I7N mice displayed significantly outer diastolic filling due to increased diastolic chamber stiffness. And but that's increase in chamber stiffness could be due to a variety of things, right? So we first thought about two things. One, alterations in post-translation modifications of the cardiac microtubule network. And the second one could be due to elevated number of cross bridges at low calcium. So here I put three citations of previous studies that they showed the importance of post-translation modifications in the cardiac microtubule network on cardiac muscle contractility and stiffness, and that's especially deterioration of the alpha-tubulin C-terminal domain. So we tested if the greater stiffness observed in the I-79N myocardium could be due to alterations in post-translation modification in the microtubule network of the heart. And although we observed significant increase in both total beta and alpha tubulin, we didn't see any changes in acetylated or detyrosinated alpha tubulin. So, okay, these findings then support our second idea, which is the greater stiffness that we observed in the I-79N myocardium at low calcium could be actually due to enhanced cross-bridge population, right? So we use measurements of sinusoidal stiffness to determine whether this variant could be influencing the overall recruitment of cross-bridge population. So this measurement, the sinusoidal stiffness, we determine this by measuring changes in sarcomere length and the respective changes in force. So be oscillating the muscle 0.2% of the original length. And we can see that the mutant myocardium actually displays an increase in sinusoidal stiffness at all levels of calcium activation, including low calcium, and that is true for both sarcomere length tested, 2.1 and 2.3. Also, when I was collecting the sinusoidal stiffness data at different levels of calcium activation, I also collected KTR. And we can see that the I-79N muscle shows significantly slower submaximum and maximum KTR. So now, how can we explain one, increase in sinusoidal stiffness at low calcium and slower cross-bridge kinetics? So what could be causing this, right? So we thought about maybe could the myofilament lattice spacing being altered in the I-79N myocardium, possibly shorter. So we went to Oregon. We had a great time. It's always a pleasure to go there. We had always so much fun. And we measured the lattice spacing, and it was not actually a surprise that we see a shorter myofilament lattice spacing in the I-79N muscle compared to the Y type, and actually both sarcomere lengths. And it was not a surprise because that actually helped us understanding the increase in active force at low calcium and also the increased number of cross-bridge at low calcium. And on top of that, this shorter myofilament lattice spacing help us understanding the slower KTR. And when we searched in the literature, we found a paper where this group did some experiments with Drosophila flight muscle and showed that the cross-bridge kinetics are slower when the myofilament lattice spacing is reduced. And 5 years ago, we published a murine study showing that the cross-bridge kinetics are actually faster when the lattice spacing is increased. So this result plus the findings in the literature can actually help us understanding what is going on, right? Next, when we look at the M3 reflection, we saw that this mutation is affecting the relative ordering of myosin heads because we observed a reduction in M3 intensity. So after seeing that, we lastly asked, could the DRX-SRX equilibrium be dysregulated? So with the help of TGA and SACT, we are able to measure the DRX and SRX equilibrium And we saw that compared to the control, the magnitude of the fast phase in the I7-9N increased significantly compared to our control. So we're talking about 84% in the I7-9N and about 70% in the control. And the magnitude of the slow phase was reduced or was lower in the mutant compared to our control. And later, when we measured the approximate percent of myosin heads occupying the DRX, we observed there is a significantly increase in myosin head population in DRX in the mutant compared to the wild type, and the population of myosin heads in SRX are actually reduced in the mutant compared to our control. So how could I explain how a mutation in the thin filament can affect the SRX-DRX equilibrium? So this is what we are proposing. So under normal physiological circumstances, there are a few DRX heads, they are checking the thin filament, searching for, let's say, available binding sites. And they are closely enough to be electrostatically attracted to the thin filament. But they will not successfully form many active cross bridges because the thin filament is actually inhibited. However, when we look at the I7-9N content, we know that this variant is destabilizing the interaction between troponin T loop region and actin, and that causes the thin filament to be less inhibited even at low calcium. So, oh, sorry, what did I do? No. Sorry, guys. Let me go back. Oh, there it is. So the DRX myosin heads, they are in the vicinity of the thin filament. They could not only form weakly bound cross bridges, but they also progress to form force generating cross bridges at low calcium. And now that we have this enhanced cross bridge formation, this could shrink the myofilament lattice, promoting now more weakly bound cross bridges. They're formed driven by the electrostatic forces that happen between actin and the nearby myosin heads. So as these heads, these DRX heads are being pulled out of the DRX heads by binding to actin, these might be expected to shift the equilibrium towards the DRX state by mass action, and that will facilitate more formation of force generating cross bridges. So here's what I would like you to take home with you. So those 3 points. One is that this I7-9N animal model, it's valuable to better understand not only the role of cardiac troponin T tail domain region in homeostasis, but also in pathogenesis. The primary mechanism by which this variant leads to myofilament dysfunction is due to destabilization of this troponin T1 loop region and actin interaction, which now we know that stabilizes the inhibited cardiac thin filament conformation. And lastly, this will contribute to hypercontractility at physiologically relevant calcium levels. And with that, I would like to thank my mentors, Dr. Pinto, Dr. Chase, all the collaborators that were involved in this project. I also say thank you to my lab members, all the funding agents, and especially the American Heart Association who funded me for a pre-doctoral fellowship, and I was able to do this project. And with that, I can take any questions. Thank you.
Thank you, Maicon, for the talk. If you have questions, please go ahead, ask the speaker.
Yeah, hi, Maicon, great talk. I already put it in a chat, just quick question. How do you explain low KTR with great amount of available cross bridges in DRX? Like usually KTR is it means a smaller KTR means less cross bridges cycling, right? Or slower kinetics.
Yeah, so slower KTR means that so KTR is the transition, how fast the cross bridge can transit from weakly bound to strongly bound. That's a very interesting question. I think the main reason why we see the KTR reduce is just because of the shrink of the myofilament lattice, which is preventing the heads to quickly transient from weakly bound to strongly bound.
Oh. So basically prolongs attached state. Okay, thank you.
Yeah. So I am blaming the shorter myofilament lattice.
Another question in the chat, you want to take?
So the question is, you didn't show any echo histology data for the I798 mice. How much hypertrophy do you see in these mice? And are there any changes in the echo parameters? Oh, that's actually a good question. Those animal models, they don't show, the I798 transgenic mice, they don't show much hypertrophy. And also they don't show much fibrosis. And when it comes to echo parameters, I personally have never done any echo data for this animal, but a previous student in our lab, she did, and she observed a reduction in cardiac output and also reduction in stroke volume. But I personally have never done echoes in these animals, but Carissa did, yeah. You showed. Oh, sorry.
Okay. No, just a quick question. In these cases, when you have a problem of activation in the absence of calcium or a very low activation level, a very good tool to detect cross-bridge formation is to check for the BDM effect, because you should absolutely take out any residual force, and so we always use this tool to check for this, because it's very common to lose activation through a thin filament problem and increase resting tension. Or...
Yeah. No, Dr. Says you have a great point. We actually thought about doing some experiments using BDM so we can exclude the cross bridge from the story and see if the increase in passive stiffness and low force can be due to some other elements. That's actually a great suggestion. We actually thought about it, but we decided to do KTR at low calcium. But you have a great point. Yes. So someone is asking if could any other factor be behind this misregulation of myosin heads. So one of the things that I see in the literature is that, for example, phosphorylation of cardiac myosin binding protein C, SRN 282, could influence the SRX-DRX equilibrium. I actually did some Western blots for phosphorylation of C protein SRN 282, but I didn't see any statistically significance between the mutant and our control. Have you seen any myofibrillar disarray in the mutant muscle? No. Someone has done some histology experiments and they didn't see any myofibrillar disarray. However, people who works with I798 IPS cardiomyocytes, they do see myofibrillar disarray. I don't know why the mutant mice doesn't show, but the IPS cardiomyocytes do. But it's actually a great point. Would you expect to see major changes in contractility if the transgenic mice show low incorporation of the mutant troponin T? I would say yes. By knowing the location of the mutation and the importance of the interaction between TNT loop region and actin, I would say that even if the mice would be incorporating less than 50%, we'll still see some significant changes in contractility. But the best way to, for sure, answer this question would be using some transgenic animal that is expressing and that is incorporating less than 50%. But my answer is yes. I think I will still see some changes in contractility for sure.
Okay. Yeah, about the myofibril disarray, so we did X-ray. We can also look at the X-ray. There's no difference. So it didn't show
Yes,
any.
our X-ray data also suggests no changes in myofibril disarray. Correct.
Mhm.